Good morning to all. Today we are going to study about the neuromuscular transmission and skeletal muscle contraction. Okay, here by studying this we can know about the neuromuscular transmission and also the role of neuromuscular transmission, how it helps in the skeletal muscle contraction. It's both of this both neuromuscular transmission and the skeletal muscle contraction is highly important. Can be asked for short essays, 5 mark questions. Okay, see, first we can see the neuromuscular transmission. What is neuromuscular transmission? Transmission of the nerve impulse from a motor neuron to skeletal muscle through the neuromuscular junction. For studying the neuromuscular transmission, first we have to know the physiological anatomy of the neuromuscular junction. What is neuromuscular junction? Neuromuscular junction is the functional junction between a motor neuron and a skeletal muscle. It is the functional junction between a motor neuron and a skeletal muscle. Okay, here we are going to study about the anatomy of the neuromuscular junction, events during the transmission, and the end plate potential, significance of neuromuscular junction, and applied aspects. First, we can see the neuromuscular junction anatomy. Here, this is the motor neuron. This white color structure is the motor neuron, and this one that is the dark color structure, that is the skeletal muscle. Here the motor neuron innervating the skeletal muscle that forms the neuromuscular junction. You can see the detailed anatomy, detailed structure. Here a motor neuron, this motor neuron, myelinated motor neuron, innervating the muscle fiber. This is the muscle fiber and this is the motor neuron. This motor neuron part that is known as the presynaptic terminal and the muscle part that is known as the postsynaptic terminal. Okay, let us see. First, we can see the structure of the presynaptic terminal. Here, this is the motor neuron, myelinated motor neuron, and the membrane of the motor neuron that is known as the presynaptic membrane. This is the presynaptic membrane. At the end of the motor neuron, at the terminal of the motor neuron, you can see the mitochondria. This mitochondria that provides the energy for the neuromuscular transmission. And here you can see the synaptic vesicles. This blue colored structure, that is the synaptic vesicles. Synaptic vesicles. Synaptic vesicles that contain the acetylcholine. And here, large number of synaptic vesicles that contain acetylcholine. That's a neurotransmitter. And here, there is an active zone at the synaptic membrane. The synaptic membrane at the junction, there is an active zone. Red colored area, that is the active zone. And here you can see here in this picture, it is not shown, voltage gated calcium channel. You can see the voltage gated calcium channel here. On the presynaptic membrane, there are the voltage gated calcium channel through which calcium influx occurs. But in this picture, it's not shown. But here there are voltage gated calcium channel. Okay, is it clear? I will tell once more this about the presynaptic terminal. Presynaptic terminal, that is the motor neuron. It's a myelinated motor neuron. At the terminal, there are large number of mitochondria and synaptic vesicles that contain acetylcholine as a neurotransmitter. And there is the active zone through which exocytosis of the synaptic vesicles takes place, and also the voltage gated calcium channel. Okay, next we can see the postsynaptic terminal or the muscle part. Postsynaptic terminal or the muscle part. This is the muscle membrane or the sarcolemma, which is known as the postsynaptic membrane. This muscle membrane forms numerous infoldings, which is known as the junctional folds or subneural folds, which actually increases the surface area for the neuromuscular transmission. And here, the space between the presynaptic membrane presynaptic membrane and postsynaptic membrane is known as the synaptic cleft. The space, this space is known as the synaptic cleft. The space between the presynaptic and postsynaptic membrane is the synaptic cleft, having a width of about 20 to 30 nanometer. Through this synaptic cleft, the synaptic vesicles are released and the neurotransmitter is released. Okay, and in here in the postsynaptic membrane, there are large number of nicotinic receptors, nicotinic acetylcholine receptors. Here in this picture, you can see that receptors. Here, large number of nicotinic receptors, acetylcholine receptor. Here you can see the acetylcholine receptor. This nicotinic receptors that is present in the postsynaptic membrane. Actually, through this synaptic transmitter binds. This is the structure of the neuromuscular junction. It's a functional junction between a motor neuron and a skeletal muscle. It consists of a presynaptic terminal and postsynaptic terminal. Presynaptic terminal is covered by presynaptic membrane, that is the neuron of membrane of the neuron, and postsynaptic terminal in postsynaptic membrane, that is the sarcomere of the muscle. Presynaptic terminal contains the large number of mitochondria and acetylcholine, and also there is a voltage gated calcium channel. Postsynaptic membrane forms the subneural folds, which increases the surface area for the neuromuscular transmission, and there are large number of nicotinic acetylcholine receptors. Okay, next we can see the transmission of the impulse. Transmission of the impulse from the presynaptic terminal to the postsynaptic terminal, which is known as the neuromuscular transmission. Here the action potential on the motor neuron. First event, that is the action potential on the motor neuron. This action potential, when it reaches the motor neuron terminal, it causes the opening of the voltage gated calcium channel and calcium influx occurs. Action potential on the motor neuron opens the voltage gated calcium channel and the calcium influx occurs. This causes the excitation of the synaptic vesicles. This causes the excitation of synaptic vesicles. And as the synaptic vesicles is excited, when the synaptic vesicles is excited, it causes the release of the neurotransmitter acetylcholine to the synaptic cleft. Okay, here you can see the release of acetylcholine to the synaptic cleft. This acetylcholine in turn binds to the postsynaptic acetylcholine receptor. Acetylcholine bind to the postsynaptic acetylcholine receptor. Here it binds to the postsynaptic acetylcholine receptor. This causes the opening of sodium-potassium channel and the sodium influx occurs. Here you can see the sodium influx. Thus, the action potential is transmitted to the muscle. Thus, the neuromuscular transmission occurs. First, the action potential on the presynaptic terminal causes the opening of the voltage gated calcium channel. Calcium influx occurs. This calcium in turn causes the excitation of the synaptic vesicle, release of the acetylcholine to the synaptic cleft, binding of the acetylcholine to the postsynaptic receptor causes the opening of the sodium influx, and now the voltage gated calcium channel opens and the sodium influx. The muscle fiber action potential is transmitted to the muscle fiber. Okay, this is the neuromuscular transmission. Okay. Here you can see the, I will show you the a flow chart for the neuromuscular transmission. Okay, here action potential on motor neuron. Action potential on the motor neuron opens the voltage gated calcium channel opens voltage gated calcium channel. This causes the excitation of synaptic vesicles. Excitation of synaptic vesicles. And the synaptic vesicles are excited, it causes the release of acetylcholine. Release of acetylcholine to synaptic cleft. Okay, when the acetylcholine is released to the synaptic cleft, acetylcholine bind to the nicotinic receptors. Nicotinic receptors on postsynaptic membrane. When this acetylcholine bind to the postsynaptic receptors, this causes the opens voltage gated sodium channels. And sodium influx occurs. Influx occurs. Thus, the action potential from the presynaptic terminal reaches to the postsynaptic muscle. Sodium influx occurs. Then end plate potential is developed. Or postsynaptic membrane is excited. Excitation of postsynaptic muscle. End plate potential developed on the muscle. Okay, you may think, what is end plate potential? Okay, what is end plate potential? That we can see. End plate potential, it is when the postsynaptic terminal is excited, binding of acetylcholine with acetylcholine receptors increases sodium potassium conductance, and resultant sodium conductance produces a depolarizing potential of about 50 to 75 millivolt, which is known as the end plate potential. Actually, what is end plate? We have seen that the structure motor neuron innervating the muscle fiber. At the point of innervation, there is an invagination known as synaptic gutter or synaptic trough. This entire structure, motor neuron with innervating skeletal muscle, that is known as end plate, motor end plate. Motor neuron ending on a muscle structure known as motor end plate, and this potential developed on the end plate, that is known as end plate potential. Okay, it is an excitatory response. Here the neurotransmitter in the neuromuscular junction, there is acetylcholine. When we study about the synaptic transmission, we can see there are large number of neurotransmitters in the different neurons, in the different synapses. But here in the motor neuron and the skeletal muscle, only one type of neurotransmitter, that is the acetylcholine. Okay. Then clinical significance of the neuromuscular junction. By blocking the neuromuscular junction, it can help the surgical operation by providing open fields and reduces movements during electroconvulsive treatment of the psychotic patients. When we do the shock, when we given to the shock to the psychiatric patients, you have seen that in sometimes in cinema, there's a shock giving. Here the electroconvulsive movement that is decreased by the blocking of the neuromuscular junction. Then other thing, that's the important thing, is the disease affecting the neuromuscular junction, that is the myasthenia gravis and the Lambert-Eaton syndrome. Both are autoimmune disease. The myasthenia gravis, muscle contraction is prevented because the neuromuscular transmission does not take place as the postsynaptic receptor, nicotinic receptor, is attached by acetylcholine type of receptors. Acetylcholine, not acetylcholine type of receptors, acetylcholine autoimmune antibodies that bind to the acetylcholine receptors. So the acetylcholine could not bind the neuromuscular, the postsynaptic receptor, and the neuromuscular transmission is prevented. In Lambert-Eaton syndrome, the calcium influx in the presynaptic terminal is inhibited. Then how the neuromuscular transmission is very, very important for the skeletal muscle contraction. Next, we can see the skeletal muscle contraction. Here, the contraction of the skeletal muscle, that is by excitation contraction coupling. Skeletal muscle contraction, that is by excitation contraction coupling. Muscle contraction. Here, this one, that is the excitation, and excitation that is followed by the contraction. Upper one, that is the excitation. This is followed by contraction. Okay. Skeletal muscle. Here, the contractile protein. Skeletal muscle contain the contractile protein as you studied. That is the actin and the myosin filament. The myosin filament, that is myosin head. This is the myosin head. Here I am just showing this to recollect your memory about the structure of the myosin and actin. This is the myosin head, which projects from the body of the myosin filament. This is the myosin head. You can see here. And this myosin head can be seen on all sides of the myosin filament except at the very center. And the myosin head have the hinges. It's a flexible portion. And this is the actin filament. You can see here the active sites. Black dots are, that is the active sites. And here this dark colored area, that's the tropomyosin, which inhibit the active sites of the actin during resting period. The tropomyosin inhibit the active sites, and there are the troponin. This is just to recollect your memory. And here you can see the myosin cross bridges projecting from the myosin filaments on all sides at 106-degree angle, and the myosin is surrounded by the actin filament. Okay, this you have to keep in your memory to study the structure, skeletal muscle mechanism of skeletal muscle contraction. Okay, here this is the skeletal muscle sarcomere, the portion between two Z filament, that is the sarcomere. The sarcomere during the relaxed state having a length of about 2.5 to 2.8 micrometer, and here the contracted state. Here you can see the difference. The myosin length of the myosin filament is same, but the actin filament here in the relaxed state, as the end of the actin filament just to begin to overlap, but here it is overlapped, and actin filaments come closer to the myosin filament. This Z filament come closer to the myosin filament. Length of the actin filament decreased. Sarcomere length is decreased. Okay. Next, we can see the muscle contraction. That's by excitation contraction coupling. Molecular mechanism of muscle contraction based on the principle that the sliding motion of filament occurs as a result of cyclic interaction between myosin cross bridges and the actin filament in presence of calcium ions. That is cyclic interaction between myosin cross bridge and actin filaments in presence of calcium ion. Okay, let us see how it works. Normally, you have seen that here there is the actin filaments, active sites are inhibited by the or covered by tropomyosin filament. Okay, this tropomyosin that is removed during muscle contraction. Okay, that is the uncovering of active sites. Tropomyosin inhibit active sites of actin during resting state. Excitation of muscle releases the calcium and the troponin calcium complex remove the tropomyosin and the active sites are exhibited for the binding of the myosin cross bridges. Okay. Okay, here you can see the active sites are exhibited. Active sites are exhibited. Tropomyosin is removed and active sites are exhibited. Okay. Then, once the active sites are exhibited, during this time, myosin crossbridge can bind to the active sites. For the cross binding of the myosin crossbridge to the active sites, first the myosin crossbridge, myosin head ATP bind and energy should be released. A sequence of events that occur during interaction between myosin cross bridges and actin molecule that is known as cross bridge cycle. Actually, what is cross bridge cycle? It is a sequence of events that occur during interaction between myosin cross bridges and actin molecule. That is cross bridge cycle. This cross bridge or myosin head attaches to the thin filaments, causing displacement of the filament over thin filament, followed by displacement in repetitive fashion. Actually, once the myosin cross bridge attaches to the active site, then it detaches, again attaches to another active site. Like this, movement of the cross bridge or myosin cross bridge along active sites of actin takes place, or sliding filament theory of muscle contraction, or walk along theory of muscle contraction. For the attachment of myosin cross bridge to the active site, one molecule of ATP is required and another molecule of ATP is required for detachment. That means two molecules are required for one attachment detachment cycle. Okay. Here you can see the normally this is the myosin cross bridge which is perpendicular in position, bind to the, comes to the active binding to the active site of actin. Once it binds, it tilt towards the center. It can tilt towards the center, producing a power stroke. Tilting of the myosin head towards the center produces a power stroke and drag the actin filament towards the center. This causes the dragging of actin filament towards the center. Then, by attachment of another molecule of ATP, it causes the detachment of myosin head from the active site. Okay, like this. Detachment. Again, it attaches to another active site. Again, it detaches. Again, attaches to another active site like this. The cross bridge attaches to active site and detaches. Thus, the myosin cross bridge move along the active sites of the actin. Here you can see the steps in muscle contraction. Before that, I will discuss one more. Actually, it's a tough part. For the skeletal muscle contraction, you have to remember about the actin and myosin, structure of actin and myosin. Active sites of actin, you have to keep it in mind. Active sites of the actin and the cross bridges of the myosin head or myosin cross bridges. Okay. Normally, during resting state, active sites are inhibited by the tropomyosin. Tropomyosin, when the calcium is released, tropomyosin is removed from the active sites and active sites are exhibited. During this time, myosin cross bridge can attach to the active site. For the attachment of the myosin cross bridge to the active sites, one molecule of ATP that bind to the myosin cross bridge. Molecule of ATP should bind to the cross bridge of the active myosin head. So by this energy, myosin cross bridge bind to the active site. You can see here. Here you can see uncovering of the myosin active sites, then the binding of the myosin cross bridge to the active site. When the binding, it causes the tilt towards the center, producing the power stroke. Then it detaches from that active site. From that active site, it detaches and ready to attach another active site. Like this, cross-bridge cycle occurs. The cross-bridge cycle, that's even sequence of events that occur during interaction between myosin cross-bridge and actin molecules. Cross-bridge attaches to thin filament, displacement of thick filament over thin filament. Okay. Let us see the steps in muscle contraction. Here it is mentioned from the neuromuscular transmission itself. Discharge of motor neuron. Discharge of motor neuron. You remember the structure of the neuromuscular junction. It causes the release of acetylcholine. Causes the release of acetylcholine. Binding of acetylcholine with nicotinic receptors increases the sodium and potassium conductance in the end plate membrane. Generation of end plate potential. Action generation, this end plate potential in the muscle that causes the action potential in the muscle fibers. Inward spread of depolarization along the T tubules. T tubules, as the action potential transmit along the T tubules, it causes the calcium from the terminal cystine of the sarcoplasmic reticulum. Terminal sarcoplasmic reticulum is highly developed in the skeletal muscle, which is rich in calcium ion and glycogen molecules. The calcium for the tropomyosin removal. Okay. Binding of the calcium with the troponin C. Binding of calcium with the troponin C. It uncovers the active sites of the actin. During this time, myosin cross-bridge active sites are uncovered. At this time, myosin cross-bridges are ready to bind to the active site with the one molecule of ATP. Then myosin cross-bridge bind to the active site, producing movement. This movement of the sliding of the myosin cross-bridge on the active sites of actin. That means one molecule of one myosin cross-bridge attaches to the active site, then it produce a power stroke, dragging of the actin filament towards the center, then detachment of the myosin head from that active site, again attaches to the adjacent active site. Like this, myosin cross-bridge walk along the active sites of the actin, which is known as sliding filament theory of walk along theory of muscle contraction. Here you can see a muscle in relaxed state, and here you can see the upper one, that is the muscle in relaxed state, and the lower one muscle in the contracted state. Okay, this muscle length of the sarcomere decreases during the contraction. The set filament come closer, actin filaments overlap. At the same time, the length of the myosin filament does not change. The length of the actin filament decreases. Okay. This is about the muscle contraction. Okay. But today we studied about the neuromuscular transmission and the skeletal muscle contraction. Neuromuscular transmission can be asked for 5 marks. Skeletal muscle contraction can be asked for 5 marks. Okay. Is it clear? Thank you.